Good point. Hi, my name is Jarek Kwiecinski. I work with Professor Diane Newman at Caltech, and my project is about the cellular response of a soil bacterium to water and nutrient stress. So we know a lot about what happens when you take bacteria and you put them in salty culture media. They lose water via osmosis, and they make these molecules called osmolytes to retain water within their cells. But dry soil is a more complex environment where cells can get trapped in thin liquid films where there's a long and tortuous diffusion pathway between those cells and a solid substrate that they need for all of their resources. So there's this question, how can a bacteria accumulate large quantities of osmolytes if it is physically separated from its nutrient supply? And that's what my project hopes to investigate, is how and when bacteria can avoid dormancy under simultaneous nutrient and water limitation. So my first hypothesis was that there is a concentration of osmolytes that bacteria must achieve within their cells that's set by the water potential of the soil, and whether or not they actually achieve that concentration is determined by the nutrients available to them in their environment. So below some threshold, the bacteria won't achieve that osmolyte concentration and will end up going dormant. Secondly, the rate at which drying occurs is very important because this osmolyte accumulation takes time. So if you have drying that happens too fast, then the bacteria won't accumulate osmolytes fast enough and will incur some cellular damage that will increase the cost for recovery, leading them to either go into dormancy or have a longer lag phase before they can resume growth in their new adapted state. So to test these hypotheses, I first had to identify which osmolytes are important to the organism that I was studying. I focused on Pseudomonas synxantha 279. It was isolated from the rhizosphere of wheat from drylands in eastern Washington. It also plays a very important role in these fields because it produces an antibiotic that kills the fungus that causes take-all, the world's most destructive wheat disease. So this bacterium is important agriculturally, and it can also be manipulated genetically in the lab. And what I did is I treated a culture of these cells with high molecular weight polyethylene glycol. These are just big chains of carbons and oxygens that are too big to get into the cells. So they withdraw water via osmosis, but don't interfere with the cell's structure and function in any other way. And what we did is, after this treatment, extracted RNA from the cells, sequenced that RNA, and then used that to identify which genes were more expressed after this water stress treatment. This graph here shows the results of that RNA-seq experiment. To the right are genes that were more upregulated, and then the y-axis indicates the level of confidence that I have in these fold change measurements. And you can see these three green dots here are the genes required to synthesize this osmolyte, NAGGN. It's a derivatized dipeptide containing 2 glutamines with some modifications. And you can see that these 3 genes for that synthesis were among the most upregulated in the entire genome. So making this osmolyte is an important part of this organism's water stress response strategy. The next thing I needed to do is develop a transcriptional reporter that will produce fluorescent proteins when the bacteria begins expressing these genes. So I can see in any environment when the bacteria is actually using this water stress response pathway. So I did that by taking the promoter that lies upstream of the operon for making NAGGN in the genome. This is a sequence of DNA that controls when the bacteria is actually transcribing these genes, and I put it upstream of a green fluorescent protein gene and then inputted that genetic material into the cell. I tested that this reporter works. You can see here that after I add PEG to cells in a 96-well plate, you can see a huge increase in fluorescence compared to the control where I added no PEG at all. So this is a good reporter for showing when the bacteria is expressing osmolyte synthesis genes. Lastly, I wanted to monitor osmolyte production over variable nutrient and water conditions. I did this in the commercial CellASIC microfluidic system. It contains a trap where cells can be held in place and you can independently change the concentrations of all the different constituents in the medium. So I could treat the cells with PEG and starve them of carbon, or ramp up the concentration of PEG over time to simulate different drying rates. This graph here shows some results from an experiment where I added PEG at 4 hours and reduce the concentration of glucose up to 100-fold. And you can see that regardless of the glucose concentration, by the end of the experiment, here's a distribution showing the fluorescence of all of the cells that I got from images of this microfluidic chip. You can see an elevated level of gene expression regardless of the carbon concentration across all of my different treated conditions that had PEG. So it seems like regardless of nutrient availability, at least thinking about glucose, these cells still make osmolytes, which was really interesting. Maybe there is no threshold, or it's a really low concentration before the bacteria actually stop using this water stress response strategy. The drying rate story was also really interesting. What I did here is I had 3 4-hour periods in this experiment where I started with no PEG, ramped up the concentration of PEG to 50 grams per liter in one treatment, or straight up to 100 grams per liter in another, and then in the slow ramp treatment, ramped up the PEG concentration to that highest 100 gram per liter level in the last segment of the experiment. And what you can see here is if I immediately bring up the PEG concentration to 100 grams per liter, cell growth completely stops. But if I gradually ramp up the concentration, growth continues almost at the same rate as if the cells weren't water stressed at all. So these cells are very adaptable. But at the same time, what's affecting the growth is not affecting the level of gene expression for this osmolyte at the level of each individual cell. Because you can see here, this is again distributions of fluorescences at the end of the experiment. So this time point here, no PEG, the fluorescence is here, the intermediate PEG concentration of 50 grams per liter. We have some value of fluorescence that's about halfway in between that and what you see for the treatments that ended at 100 grams per liter, whether that was a fast or slow ramp. So they still achieve the same level of gene expression, they just grow differently. So maybe there's some damage that the cells incurred with the rapid treatment that just requires, there's additional things that the cell has to do to keep growing to repair some of its machinery that got damaged during that transition. So where I'd like to take this, I'd like to study these processes in actual unsaturated porous media. We have a micromodel in my lab where you can, here I have an image of cells and air and these solid pegs. It's intended to mimic the porosity of sand, so it would be really cool to see if I can visualize water stress responses in these actual thin liquid films. And I also think it would be cool to get a sort of 1D steady state nutrient concentration profile between where cells are located and where there's solid substrate, thinking about extracellular enzymes and the diffusion of the dissolved solutes that these enzymes release. So that way I could use this concentration profile as boundary conditions for further experiments to kind of bring this into the environmental context. Thank you for listening.